Hello everyone, my name is Bruno Lima and I will present you an HLA Evolutionary Divergence Calculator. So the two classes of proteins with the highest degree of polymorphism in the human genome are human leukocyte antigens class 1 and class 2. The polymorphism of HLA is not only one of the main contributors for humanity diversity, but with a genetically predetermined history, HLA have a key role in the immune response. The HLA heterozygous advantage hypothesis states that heterozygous genotypes for HLA loci are more likely to trigger an immune response in the presence of an infection. This hypothesis of heterozygous advantage has been extended considering the sequence level, generating the notion of divergent allele advantage. The divergent allele advantage hypothesis stipulates that diversity between HLA alleles of the same locus may increase the functional capacity of peptide antigen presentation and as such increase protection against pathogens and tumors through the ability to activate protective immunity of T cells. The HLA Evolutionary Divergence (HED) score has been proposed as a way to quantify this divergence between alleles of the same HLA locus. So an individual with an higher HED value between HLA alleles may allow the presentation of a more diverse immunopeptidome. Here I will present you a simple application that allows the calculation of this HED score. The HED value is calculated using the Gratham distance, which estimates the range of the immunopeptidome provided by the molecules encoded by a given HLA locus based on the difference of paired chains of amino acids for two HLA alleles of the same locus. This application was built with Shiny in R and using the package stock for HED computation. Here you can see this simple application where only inputting the HLA alleles of each locus, you obtain the respective HED value. You can do it for class 1 loci and also for class 2. Here, as you can see, each time you select different alleles, you obtain the respective HED. Also, you can input a file with multiple genotypes. Here, an example where you provide an ID and the HLA genotype for the key main loci in HLA, and as a result, you will have the respective HED values. Let's upload an example file. Just browse it, and that's all. The only thing you have to assure is that the column names are equal to this one that I'm showing to you. So these are the values, the genotypes you input, and as a result, here you have the 5 HED values, HED columns with HED values. Another thing, none of these results are saved anywhere, so the data complies with the data protection rules, and here the application only do the calculations, and you can download the results to your personal computer. Also, we'll mention that this application is part of the Transplant Open Registry initiative, and here you can find other applications related to histocompatibility tests and also related to transplantation. The HED value is computed with through an R package, the estoc R package, and mainly with the simple function where you input the two alleles of HLA locus, and as a result, you have the HED value. So, as a conclusion, we can say that when we have an HLA genotyping with a two-fold resolution, we can have an HED value without incurring on any additional costs. This application is open source and freely available to everyone. Nowadays, we can find already several studies describing HED association with cancer treatment, immunotherapy approaches, outcomes after cell transplantation or even organ transplantation, and even with HIV control and COVID-19 outcomes. So hopefully this app will be a useful tool to those researching the link between the allele advantage and certain disease. And that's all. Thank you for your time.